We're going to start by going to the RAST website and setting up an account. So the RAST URL is rast.nmpdr.org. And in order to sign up for an account, down here at the bottom, you have to register a new account. When you do this, you can enter in your name, your first, last name, email, etc. And this request will go to a person for approval, so it may take a few moments. So I will try to set up a new account. I'll make my account name match my name. My organization is Argonne. My country is United States. And I don't have a group, so I will request. So after the request has been approved, you'll receive an email. The first email that I got looks like this, and is basically a polite reminder saying that they will be approving the account. After that, you'll get an official email enabling you to set up the account. The second email will have your username and your password and a statement saying that your request has been accepted. So I will remember that and go back to the RAST website and log in. Now, the first thing you should do after you've logged in is change your password to one that you can remember. So over here on the right hand side, there's a little icon that looks like two people. If you click on that, it might take a moment. It'll bring up your personal settings, and I'm going to change my password. And we will change the password by clicking Perform Changes. So it now says you have successfully changed your password. So I will go back to the Home tab. At the Home tab, you can scroll down through here and you can see different links to tutorials, how to register and so forth, tutorials for how to improve annotations and so forth. We want to go to the jobs overview . This is where all of your jobs will show up after you've uploaded and submitted them. If you notice across the top here, there are currently 58 jobs in the RAST queue. Depending on the queue size, that can affect how long it takes for your genomes to be annotated. And so sometimes it's advantageous to watch this and submit an annotation job when the queue is smaller. So in order to follow along, I think that it's probably a good idea for us to download a genome of our own and try. So I'll open a new window. In order to demonstrate how to annotate with RAST, we'll go back and bring up a new browser window, and we're going to download the E. coli K12 config from NCBI. So in your browsers, in the browser window, we will type in, and this is a long URL, so I'll go slow, ftp:// ftp.genome ncbi.nlm.nih.gov/lowercase genomes/bacteria with an uppercase B /capital E Escherichia SCHERICHIA underscore coli underscore K underscore 12 underscore SUBSTR underscore capital MG 1655 underscore UID 57779 /NC 0000913.FNA. This is the FTP link to the E. coli K12 chromosome. So then hit return. Now, you can see the chromosome, and what we're going to do is we're going to download it. And each browser is different, but I'm in Chrome, so I'm just going to save page as. And that's going to bring up a saving window. And I'm going to leave off the .txt extension because I'm in a Mac and it likes to add .txt to everything. And then I'll click Save. Now I will minimize this screen and bring up RAST again. Okay. Now that I've saved the E. coli K12 config, I'm going to come down here on the jobs page. Oh, the queue has shrunk, which is really nice. And we will upload a new genome. This will bring us to an uploader page. And under here, it says sequences file, and we'll choose a file. Now, I placed that in my documents. Here's our file. We'll open it. And we will click Use this data and go to step 2. And it'll show you the status of the upload. This may take a few moments depending on the size of your genome. It's important to note that RAST currently only supports bacterial and archaeal genomes. So you don't want to try to upload a eukaryote because it'll probably break. Now, it'll compute some initial statistics on the genome that you just uploaded. So this is E. coli. It's 4.6 MB, 50% GC, etc. And in order to annotate it, we have to insert a certain amount of metadata. Now, E. coli K12, I happen to know its taxonomy ID, so I'll enter it. But in most cases, you may not know this. Now, the taxonomy for E. coli K12 is 83333. And if you know the taxonomy ID from this is the number from the NCBI taxonomy browser. It will auto-populate the taxonomy string, and it'll fill in the scientific name, domain, and genetic code for you. If you don't know it, then you have to go back through and tell it whether it's bacterial, archaeal, or virus. Give it a genus name, a species name, a strain if you know a strain, and a genetic code. People always ask about these genetic code numbers. They are the NCBI genetic code numbers. 11 represents most bacterial and archaeal genomes, and 4 represents the mycoplasmas and a few other things. This is an important bit of metadata because the gene calling algorithms are looking for stop codons, and the mycoplasmas have co-opted one of the stop codons for tryptophan. So it makes a difference in how the genes get called, which is why we require it. It's also important to point out that with the genus and the species name do matter in the more advanced use cases. So although you can type anything here, you're far better off to type in something that's reasonable, because it can affect the downstream decision making in which programs get run in the advanced use case. So now we're going to click on Use This Data and go to step 3. For this initial demonstration, we're going to use classic RAST. We've recently done some work that enables you to use the RAST toolkit, which is the RAST TK option, and I'll demonstrate that later. But we will use classic RAST. The gene caller is RAST, but what that really means is that you're using Glimmer 3 with some preset options. You'll select the FigFam version. We always recommend using the default in this case, unless you're very adamant about making new sequences look like old new annotation jobs look like old annotation jobs. Because the releases have been a steady improvement. You want it to. We will leave the checkboxes set as the defaults. You're welcome to explore how these may work, for instance, fixing frame shifts and so forth. And then we will click Finish the upload. After your job is uploaded, you will get a job ID, and you can view the job status or go back to the main upload page. If you end up getting lost, you can go to the jobs overview under the tab here. And now under the jobs overview tab, you can see that I've just submitted one job with the ID 217983. It's E. coli K12. It's been assigned. This is the 80th time that E. coli K12 apparently has gone through the system with someone who has chosen to give the proper taxonomy ID. It has one contig, it gives the size, and then over here you have a progress status. And it looks like we're currently waiting in the queue, so we can click on the View Details. And if you click on View Details, it shows you information about the particular job that you submitted. After the job finishes, some of the information in this table will be filled out. Now it's just a matter of waiting to see for the job to actually complete. Okay, now I'm going to demonstrate how to run the RAST TK pipeline from the RAST website. So we need to go back to rast.nmpdr.org. And the last time I left, I never logged out. Otherwise, you'll have to log in. And we will want to upload a new job. Now we're going to upload that same E. coli K12 genome that we originally annotated with classic RAST. So we will choose that file and we will upload it again. Then click Use This Data and go to step 2. The RAST toolkit is an exposure of all of the programs that are running, that RAST currently runs. We have recently exposed all of the tools so that users can attempt to create their own annotation pipelines. So now that the genome has been uploaded, we're back at the original first page, which is for inserting metadata. So we will proceed through this just like we did before, entering our tax ID for simplicity. Again, if you're doing your own genome, you may have to enter this by hand, and then we'll go to step 3. When you get to step 3, instead of classic RAST, you're going to want to choose RAST TK. And if you don't check anything else, this will run the default RAST TK pipeline. But the RAST TK pipeline is now exposed, so if you click Customize RAST TK pipeline, it will bring up a table where you can customize the steps that you want RAST to run. So all of these individual rows represent a program that RAST is running and the order in which it's running them. And the checkboxes means that a particular program is turned on. An unchecked box means that it's turned off. You can also experiment with changing the order in which programs are run by clicking on a row and dragging it over another row. So in this case, now we'll be calling pyrolysines before selenoproteins. Usually, there are situations, in most situations, this doesn't matter. But when you get down to the particular algorithms that are actually annotating rather than calling features, this can become an important feature. Now, since we want our new annotation to differ from the one that we just submitted in the classic pipeline, let's turn on calling insertion sequences and searching for prophage elements with PhySpy. Finally, all of the remaining submission steps will be the same. And before we submit, I want to point out that you can change conditions and parameters on the right side over here, but users typically would not want to do that. Finally, we will click Finish the upload. This will submit the new annotation job to the same RAST queue, and a new job will show up on our jobs overview page. So now, the RAST TK job, which is will come in as the first cell in the row. And so now I have two jobs waiting in the queue. The first one, the .81, is the RAST TK job, and the .80, which is the previous one, is the classic RAST job. Now we'll wait for both to finish, and when they finish, we will get an email from the RAST server. Okay, it's been about a half hour, and I've gotten my genome back. I've gotten an email from the RAST server saying that our annotation job is done. So we're going to go back to RAST, to the RAST website, and we're going to log back in. Oh, I already am logged in. And we'll go to the jobs overview page. Now we'll be able to see that the annotation progress is complete on both the RAST TK job that we submitted as well as the RAST Classic job that we submitted. So the first thing I'm going to show you are results from the RAST Classic that we did first. So I'll click on the View Details link beneath the annotation progress. At the very top, there are several links for actions that you may wish to take on this genome. The first is you can browse the genome in the seed viewer. I'll come back to that. You can also download the job in a variety of formats. So amino acid FASTA, GenBank, the Excel formatted spreadsheet, etc. You could share it with others if you know their email. And you could also view it in the context of its phylogenetic neighbors by clicking on this link that says View Close Strains for this job. This tool, we commonly refer to this as the What's Changed tool, and that's a subject of a tutorial on its own. If you keep scrolling down, you see information about the job. You'll see here that we ran the classic RAST pipeline on it, and whether you fixed frame shifts and so forth. You'll see the number of features that were called. In this case, there was 4,554. Now, a feature could be any sort of DNA element. In this case, with the RAST Classic, it refers to RNA genes and protein encoding genes. So we'll scroll back up and we'll click on Browse Annotated Genome in the seed viewer. And this will bring us up to a new web page for the seed. The seed is the database that contains the annotation information that RAST commonly accesses. And it also provides an environment for annotating. This will bring us to an organism overview page with information on the size and the number of coding sequences, subsystems, RNAs. A subsystem is a set of logically related protein encoding genes that are related by function. And then there are links here for comparing your genome to others, downloading your genome, or proceeding into annotating it. If you scroll to the bottom, you can see information about what the actual processes were that the functions that it actually found as they pertain to the subsystems. And so you can see they're color-coded in the pie chart here. We can click on Browse and browse through the features of E. coli K12. And this will bring up the actual genome and you can page through it. Or if you were to click on the first protein encoding gene down here, this link, it'll take you to the protein overview page for that protein. And if you scroll down, it will load the Compare Regions tool. So when you get to this page, the protein page, you can walk the genome and you can see information on the annotations and its comparison to other organisms. So here in the protein comparison page, you can see the first gene that we called in our genome, and you can see close relatives. So here's Salmonella and Serratia and Yersinia, etc. And you can see the conserved chromosomal order. And if you do a mouse over, you can see what these proteins are. So this is threonine synthase number 3 that I'm rolling over. The color refers to how similar the proteins are based on BLAST. So all of the orange ones are homologous. And using these buttons up here, you can page through regions. And so I'll click that once, and it should take you to the next window. As that's re-rendering, if you come up along here, you can see that you can edit a functional role, and you can see the annotation history for why we think this first gene is aspartokinase. So if you click on that, I'll click on it here. You can see what tool was used to create that. So here's the next region in the Compare Regions tool. So it's jumping up here by 1600 base pairs, and you can change the window size, and you can also change the number of organisms that the Compare Regions tool is bringing in. So this is how you walk your genome and compare it to other organisms in the seed. Now, I want to go back to the other job and show you some information about it. So if I go back to the jobs overview, so I just closed the tab that had taken us to the seed. So I'm going to close the jobs overview, or go back to the jobs overview rather. And now I'm back at the point where I have my 2 annotated genomes, and I'm going to click on the View Details page for this one. Now I want to attempt to download this genome. Remember, these were the same genomes, but we used the RAST toolkit for this one as compared to the classic RAST algorithm for the other one. And we have more features now because the RAST toolkit contains software for finding CRISPRs, which are called as features. And remember, we also found insertion sequences and other things like that, and prophage. So we're going to have more features come out here. If we download it, I'm going to download it in Excel format. You simply select which format you want and click download. And this will bring us up a spreadsheet. Here we go. And the spreadsheet will have the config ID, which is the same for each one. The second column is the feature ID. In this case, whenever you call with the RAST toolkit, you'll see that different features get called different things. So here, for instance, we have, instead of a PEG, which is shorthand for protein encoding gene, there we have a repeat. And if you scroll down, here are some RNAs. Here's some more repeats, etc. You could sort this column, and for whichever, if you've sorted this column alphabetically, you could sort out the features that you cared to look at. And it also contains information on the location, the start, stop, and the function that it was called. If you scroll over even more, it has evidence if there was a FigFam associated with it. And then the very end of the table contains the nucleotides sequence, and if you go the whole way over, you get the proteins. So this is just one format in which you can download your data that people find convenient. So this concludes our brief tutorial on how to get started annotating a genome using the RAST website and the RAST toolkit. We only briefly touched upon some of the more interesting and powerful tools that are available through RAST and the seed, which should be a subject of another tutorial. But this video hopefully is enough to get you started so that you can start annotating on your own.